Hi students, good morning to everybody. Today, the 11th classes, which topic is going to be classified as transport and plants. In transport and plants, today which topic to be going discuss about is commonly called as source and sink. So what do you mean commonly called as source? Source is nothing but any part of the plant or any organ of the plant which is capable for exporting food to the regions of metabolism, or to the regions of storage, that is commonly called as sources, which things especially utilized for the process commonly called as example matured leaves and germinating seeds. And next to be called, what do you mean commonly called as sink? Likewise, any part of the plant or any organ of the plant which receives food materials from source, that is commonly called as sink. In plant, what are the part it must be act as a sink is commonly called as roots, tubers, developing fruits, and immature leaves. So for here, source is a production regions. Sink is commonly called as utilizing regions. After that, which topic is going to discuss about is commonly called as phloem loading. Phloem is nothing but some kinds of complex tissue. It is especially transport food materials. So what are the definitions of phloem loading? That means which process it is generally manufacture of food. That process commonly called as photosynthesis. So photosynthesis is a process That's manufacture of food. That food materials translocated from mesophyll cells to phloem sieve elements in the mature leaves. So what do you say? Phloem loading, that means which way plant responsible manufacture of food is commonly called as by the process of photosynthesis. That's what we say. Photosynthesis is the main process manufacture of food. The kinds of food live from mesophyll cells to phloem sieve element in the mature leaves. It consists of three ways. The first one, sieve tubes conduct only sucrose, but the process of photosynthesis is manufactured. The food is commonly called as starch, or also known as triose phosphate. The starch and triose phosphate, which organelle is manufactured is a food, means chloroplast. From chloroplast, the starch is translocated into cytoplasm. In the cytoplasm, the starch and triose phosphate is converted into sucrose. Why the starch became converted into sucrose means for further translocation. This is the first objects. Second object is commonly called as sucrose. Leave from mesophyll cells to Neyer's elements by short distance transport. This is second thing. Third thing, from sieve tubes to sink or from sieve tubes to sink by long transport distance. So those three objects comes under phloem loading. Can I go to the third one is commonly called as phloem unloading. What is the meaning about phloem unloading? So already the process of photosynthesis, the food materials are stored. There's a commonly called as phloem unloading. What is the phloem unloading means? From C elements, sucrose is translocated into zinc organs. What are the things to be act as a zinc organ? You know that roots, tubers, developing fruits, and young leaves. The phloem unloadings also it can be consist of three ways. First thing, sieve element unloading. That means sucrose is leave from mesophyll cell. These are first things. Second thing, short distance transport. That means the movement of food from mesophyll cells to zinc cells. These are commonly called as short distance transports. Third one, storage or metabolism. That means these are commonly called as last steps of phloem unloading. What are the process to be happen in the last step is commonly called as the storage of sucrose became utilized for various purposes. Which purpose are generally it can be utilized means for transporting food for various organs. That's the reason the zinc cell receive food from mesophyll cell. It carry out storage and the metabolism. So it can store. After the stored material, it must be going on metabolic process. What is the commonly called as metabolism? The existing two process, anabolism and catabolism. That means building up process and the breaking down process. Those process is always accompanied with zinc cells. Can I go to next one discuss about the topic is commonly called as mechanism of translocations. How the mechanism it must be takes place. Based on the way, there are several hypotheses can be explained. But all hypotheses, it is related to translocations of food. Some of that hypothesis now can discuss about. The first one is commonly called as diffusion hypothesis. So what are the commonly called as diffusion? The movement of molecule from higher concentration to lower concentration based on the way translocation it must be takes place. That is commonly called as diffusion hypothesis. So here the movement of material, what way it must be going on, or otherwise known as transporting of food materials from high concentration to lower concentration, or the movement of food materials from higher concentration to lower concentration, which is it must be considered as a higher concentration means place of synthesis. That is commonly called as, it may be possess high concentration. It may be, what is the commonly called as low concentration means place of utilization. So the food material, it must be moved from high concentration to lower concentration through physical process. But the theories in general is rejected. Why the theory is generally rejected and commonly called as the speed of translocation is much higher than simple diffusion. So that is a real fact. The speed of translocation is much higher than the simple diffusions. In addition, the translocation, it is not a physical process, it is a commonly called as biological process. That is the reasons also the theories become rejected. So what we say? The translocation is not a physical process, it is a biological process. So any biological process, the process became stopped by influence of any poison. This is a commonly called as diffusion hypothesis. That means the movement of food from the regions of higher concentration to lower concentration through physical process. But the theory is generally rejected. What is the reason is a commonly called as the speed of concentration of food is much higher than the recorded simple diffusions. In addition, diffusion is not a physical process, it is a biological process. That is the reasons also they can be rejected. In addition, biological processes, it may be stopped by influence of any kinds of toxin which may be present. Can I go to the second one theory is commonly called as activated diffusion theory. The activated diffusion theory, who are first given that word is commonly called as Mason and Maskell in the year of 1936. According to activated diffusion theory, that means diffusion in sieve tubes what do you mean? Diffusion in the sieve tube is accelerated, that means speeded, or who responsible speed the process. So diffusion in the sieve tube is accelerated given by activation of diffusion molecule. So through activation of diffusion molecule in the sieve tubes, the movement of food materials from higher concentration to lower concentration or decreases protoplasmic resistance in the diffusion. So those two is responsible for carry out activated diffusion theory. Can I go to the third one is commonly called as potential theory. What I call potential theory is nothing but the movement of substance, it must be takes place based on osmotic concentration. These theory who are responsible first to demonstrate commonly called as Fenzer and Spanner. Fenner when the year is 1957. Spanner is commonly called as 1958. According to this theory, the osmotic potential across the sieve plate, that means the osmotic potential across the sieve plate which cause movement of water along with the solids. Movement of water along with the solids. This theory also became rejected. Why means the Fenner-Spanner could not proper explanation given by mechanism of translocations of dissolved substances. That is the reasons also this theories also became rejected. Can I go to the last one theory is commonly called as Munch mass flow hypothesis. Munch mass flow hypothesis who are first given by Munch in the year of 1930. But the Munch could not given a proper explanation. But later, the greatest scientist commonly called as Crafts in the year of 1938 to give proper explanations of mass flow of hypothesis. So according to this theory, organic substances are solute move from high osmotic pressure to low osmotic pressure along with Turgor pressure gradient. So this hypothesis is based on organic substances or solutes. It must move from higher osmotic pressure to lower osmotic pressure along with the Turgor pressure gradient. How it can be moved? Based on the observation, some experiments to be proved. What type of experiment to be utilized to prove NMAS or mass flow hypothesis mean? Can I give one like this? This is a beaker. The beaker containing water, also called a reservoir. For the experiment, you got to utilize two columns. That means column A, column B. To prove NMAS moving off solute, for the experiment to be given like this, commonly called as column A, you have to fill it with high concentrated solution. Column B, they can be possess dissolved solute. So the A column and B column, which is responsible for connect or joint, is a commonly called as tube. The tube is commonly called as T-tube. So the T-tube that's connect tube A, that means column A and column B. Column A, they can be possess high concentrated solution. Column B, they can be possess dissolved solutes. This is a setup you have to be keep under seconds for some hour. After some hour, if you are keeping that apparatus pin, what are the changes are there? You have to be observed. What? The first stages. So after that, the observations given one, the first one is commonly called as high concentrated sugar solution is always occur in the form of hypertonic state. Due to hypertonic state, the water molecule drawn from the reservoir so due to the hypertonic states, the solution A come out or absorb water from the reservoir. This is a commonly called as the first step. Second thing, which way a column A or solution A receive water from the reservoir is commonly called as by the process of endosmosis. So through the endosmosis, maximum amount of water it must be enter in the solution A, resulting created turgor pressure. This is the second. So how is the turgor pressure created? Continuous entry or ionized water entry into the solution A or column A, resulting turgor pressure is created. Third thing, increasing turgor pressure what is that? Increasing turgor pressure, solution A from the solute it is moved to reach solution B through T tube. That's it. The process commonly called as what? Increasing turgor pressure. So what I am to say, increasing turgor pressure in A solution, the solute molecule it must be moved from solution A to solution B through T tube. That is commonly called as the third objections. Third observations. Fourth one, for continuous entry of solution from solution A to solution B means at the end of the nature both solution A and the solution B became is isotonic solution or isotonic condition. How it is make isotonic solution? Because continuous flow of solute from solution A to solution B at the end of the nature solution A and solution B became is make regarding equilibrium, it make isotonic solution. When solution A and solution B it must be present at the isotonic solution, the flow of solute became it will be stopped or the progress will be stopped. When the process became to water continue, that means a small amount of sugar solution add in the solution A. That is a phenomenon. So both A and B is equal solute nature means the process will be inactive or stopped. The process to be again continue means a small or a pinch amount of sugar solution that must be added in the solution A. Then again the process will be going on. Why that experiments to be given for you mean such a same process is implemented in the plants also. How means? Which is in plants act as regarding what A? Solution A, it is analogous to mesophyll cell. What is say? Solution A, it is similar to mesophyll cell or analogous to mesophyll cell. Why means? Mesophyll cell which containing high amount of food in soluble condition. That food materials leave from mesophyll cell which is leads to reach on that phloem elements because the phloem is commonly called as storage of food materials. So that is a or solution A, it is analogous to mesophyll cell. It can be containing high amount of food in insoluble condition that is considered as a source. Likewise, solution B, it is analogous to cells of root and stem. The cells of root and stem, what is that real phenomenon is commonly called as receiving food materials from source. So will be considered as regarding it is a sink. So B, solution B, it must be considered as sink. Likewise, T, it is analogous to phloem tubes. It is analogous to phloem tubes. Now come to the conclusion like this. Here, mesoble cells drain water from xylem elements. What do you say? Mesoble cells obtain or drawn water from xylem elements by the process of endosmosis. By the process of endosmosis, the water molecule is obtained in the mesoble cell so that increasing turgor pressure in the mesoble cell. So what do you say? Mesoble cells draw water from xylem, resulting increasing turgor pressure in the mesoble cells. At the same time, cells of root, cells of stem of the phloem, comparatively in that mesoble cell, the turgor pressure is much lower. So if the mesoble cell turgor pressure is higher, the cells of stem and the root the mesoble is comparatively is very lower. That's why insoluble food materials leave from mesoble cells to phloem, or the soluble component of food materials leave from mesoble cells to phloem cells of stem cell as well as root cells. There, the root cells and the stem cells utilizing such a kinds of food or remaining food is stored in the form of insoluble condition. During the condition, some amount of water is releasing. That releasing water is to go out regarding xylem elements through the capillary. Can I go to the next one? What are the merits of mass flow hypothesis? That means woody plants or herbaceous stems, you are going to griddle. Griddle means removal of bark. Where you have to be making griddled from that place, high concentrated solution is exudate from that place. This is the first merits. Second merits, positive concentration gradient is disappear. How the positive concentration gradient disappear? Why it can be lost? Because the plants are going to defoliate. What do you mean commonly called defoliation? Removal of leaves. These are two is the commonly called as merits. Then what are the called demerits? First things, based on the theory, the translocation of solute is only unidirectional. But Some other plan, the translocation of solutes is not only unidirection, it also carry out I-direction. That's why the theory is become rejected. Second thing, osmotic pressure in mesophyll cells and root have not possess requirements. So proper requirement is not occur for the osmotic pressure in mesophyll cell as well as root hairs. That is the reason also rejected. The third one, the flow of solutes in sieve tubes and protoplasm in passive manner. What do you say? The flow of solute in sieve tubes and protoplasm is passive manner. That way given explanation in mass flow hypothesis. But some of the scientists to explain the flow of solutes to require some amount of ATP. So these are things to be considered as merits of mass flow hypothesis and demerits of mass flow hypothesis. Thank you.